Hi ladies, this is the video on protein synthesis about transcription and translation. So before we start, what exactly is a protein? This is the first thing you need to understand. So basically a protein is a macromolecule made of amino acids. So amino acids are the monomer, they're the building block of a protein. So they're like so if you think about like Lego blocks, those little 4 square Lego blocks. When you add them together, you can make a big structure. Okay, that's like the macromolecule, that big structure. But the individual Lego blocks, those are like the building blocks of that protein. So they're like the amino acids. And what's a protein used for? Well, proteins are used for structure. So like something like keratin that's found in your hair and fingernails, or elastin. This is what makes your skin stretchy. So like when you can like pinch your skin and it kind of like pulls, it's because of the protein elastin. Or enzymes like DNA polymerase that adds nucleotides to make a new DNA molecule. Transport, remember the sodium-potassium pump is involved in transport of sodium-potassium across a membrane, which is really important for the nervous system. Something like a messenger, so the growth hormones, or something like insulin. So insulin is really important to regulate your blood sugar level, or something like an antibody in the immune system. So all of these things are really, really important. Proteins are probably the most important molecule that we're going to learn about this year. This is what an individual amino acid looks like. For more specific different amino acids, the R group is what's changed out to make the different kinds of amino acids. So when they go together, they look like something like this, and then they would have a specific structure for something like glycine or isoleucine that makes them a little bit different. Note the three-dimensional structure of this protein right here, and remember that 3D structure is really, really important to determine the function of a protein. Okay? If the structure, the shape isn't correct, the protein won't work properly. So for example, if you have maybe insulin isn't correctly formed, okay, it's not going to be able to regulate your blood sugar level. Or if you have an enzyme like DNA polymerase, okay, if it's not correctly formed, okay, it's not going to be able to add those nucleotides. Remember, the shape determines the function. You can think about that toothpick ace whenever you had to tape your fingers at the beginning of the year, and when you couldn't use your fingers, it was a lot harder to carry out that reaction. Okay, so 3D structure of protein is really, really important. Okay, so how are proteins made? Proteins are made through transcription and translation. Transcription is the process of creating mRNA from a DNA template in the nucleus. Translation is when the mRNA is used to make a protein at the ribosome with the help of tRNA. So before we get going, I'm just going to do a quick comparison of RNA and DNA. I mentioned a little bit about RNA and DNA here, but I just want to kind of show you a little bit what the differences are. So this is where you're going to be filling in that little Venn diagram. So number one, both RNA and DNA are both nucleic acids. Okay, they are made up of nucleotides. So they are nucleic acids. The building blocks are nucleotides. However, the nucleotides are a little bit different. So you can see here in RNA there is ribose sugar. So ribose sugar makes up a nucleotide. In DNA, remember, you have a nitrogenous base, you have a phosphate group, and then you have deoxyribose sugar. Okay, so they still have the same groups. They have a phosphate, they have a sugar, and then they have a nitrogenous base. The sugar is just different between RNA and DNA. Whenever we also look at their 3D structure, they're a little bit different. So for one, RNA is typically single-stranded. Okay, DNA is double-stranded. Okay, so it's a double-stranded double helix, or the double helix, that's double-stranded. Okay, so you have one strand on this side, one strand on this side. They go together and make that twisted ladder. RNA is single-stranded. Another thing that's a little bit different, in DNA, remember we have cytosine, guanine, adenine, and thymine. Those are the nitrogenous bases. Remember the complementary base pairing between the different nitrogenous bases that are held together by hydrogen bonds. And those hydrogen bonds can break to replicate the DNA. Well, RNA is a little bit different. It is single-stranded, so there aren't any hydrogen bonds, and there is one different base. So you have uracil now instead of thymine, but it is still used in a very similar way. So you have, whenever you're making your mRNA copy, cytosine will pair with guanine, and adenine will pair with thymine, and then uracil will pair with adenine. And I'll show you how this works in a little bit. Okay, so these are the types of RNA. I mentioned them before. Messenger RNA, or mRNA is what we call it, is really important. It basically carries the DNA, the information in the DNA out of the nucleus. Okay, it's a copy of the DNA. So you know how like sometimes books in the library, you're not allowed to take them out of the library, but you can make a photocopy of them? The mRNA is basically like a photocopy of the DNA. Okay, and this is going to carry the information to the ribosome. Okay, so here we have tRNA and ribosomal RNA, or rRNA. The ribosome is basically it's basically the way that a protein is made. So ribosomes are little protein factories, and it's going to use the mRNA template and help assemble the new protein or amino acid. Okay, transfer RNA, or tRNA, is basically the little taxi that carries the different little amino acids to the RNA. So it's it goes out and finds the correct amino acid, and it brings them to the ribosome to help assemble and make that protein. Okay, so let's get into transcription a little bit more. And remember that this happens in the nucleus. Okay, this is the photocopy process. So this is what happens whenever DNA is used as a template, made a copy of it with mRNA. So the DNA molecule is going to unwind and loosen up a little bit. RNA nucleotides are going to come in, and remember that complementary base pairing takes place. So C compare with G, G compare with C, U with A, U with A, C with G, A with T. Okay, complementary base pairing, just like whenever DNA replicates. But what happens what's really great about the mRNA copy is that it doesn't stay permanently attached to the DNA. It just kind of copies, makes that little photocopy, and it can be taken out of the nucleus and out of the cell. So this way the DNA stays protected, it doesn't get messed up, and the RNA can take that message to a place where a protein can be created. Okay, so this RNA transcript, or mRNA, is going to leave the nucleus and go find a ribosome. This is the process of translation. This is going to happen in the cytoplasm at a ribosome. Before I get started, one important thing to know is that there's a triplet code, or like codons, that are really, really important for how this process takes place. What's really, really interesting is that all organisms on our planet use the same code. We have the same genetic code. We have the same 4 nucleotides. Really, really important that we have that. So everything kind of works the same. We have the same enzyme, same ribosome, same way of making proteins from DNA. Okay, so remember that the mRNA is a copy of the DNA, okay, and then the triplet code is that 3 of the mRNA nucleotides are going to code for 1 amino acid of the protein. So one codon, okay, one group of 3, one triplet, is going to code for 1 amino acid of the protein. And this is really important to understand for translation. And this is a codon table that basically shows you which amino acid corresponds to which sequence of nucleotides in the nucleotides in the mRNA. We're going to practice this a little bit more next week. Okay, so let's go through and see how this works. So here is a tRNA molecule. Note that there is an amino acid attached to it, because these are the little taxis that bring the amino acids, the little building blocks of proteins, to the ribosome. Okay, the tRNA can match up with a codon of the mRNA. Okay, we call it the anticodon and the mRNA, and this will bind, okay, and bring the correct amino acid at the ribosome. So this is an important piece of information to know in a second. Okay, so here's the ribosome, and this is how this is going to take place. So basically, you see the ribosome, there's a large subunit, there's a small subunit. The mRNA kind of feeds in between them, And the sequence of nucleotides on the mRNA is going to tell the ribosome what the correct tRNA is that will fit and bring the correct amino acid in. So let's look at how this takes place. So basically, so you have tRNAs with amino acids that are floating free in the cytoplasm. The correct tRNA will link to the correct amino acid, and it will transfer this amino acid to the ribosome. Okay, so it's going to come in, and it's going to bring the appropriate tRNA. The appropriate tRNA is going to come in and match up to the mRNA codon at the ribosome. And from here, the sequence of all these tRNAs bringing in the correct amino acid, these little amino acids can get bonded together and they can create a polypeptide chain. Okay, so let's look at this in detail. So protein always starts with the start codon. The start codon in mRNA is AUG, and that codes for methionine, which is the first amino acid of every single protein. So the methionine is going to come in, and it's going to bond to that anticodon is going to bond to the start codon on the mRNA. This will signal the large subunit to attach. And what's going to happen next is that the next corresponding tRNA molecule is going to match up the anticodon with the codon, and it's going to bring in the appropriate amino acid. A bond will form between those two amino acids, and this will cause a little shift, in the mRNA, and it'll make room for a new tRNA molecule to come in. So a new tRNA molecule will come in. It'll attach the anticodon to the appropriate codon, bringing in the correct amino acid, and this amino acid will form a bond. So there'll be a bond formed between leucine and tyrosine, and this will allow a amino acid to be formed. And this will continue and continue and continue until there is a chain of amino acids. Okay, so let's look. So once all these amino acids are grouped together, it will eventually stop when there's a stop codon, which basically the stop codon is a tRNA molecule that doesn't have an amino acid attached to it. And this will form a chain of these little amino acids together. What's going to happen next is this chain needs to be folded appropriately so it gets its proper three-dimensional structure. The three-dimensional structure, remember, determines the function, so it's really important that it's folded correctly so it functions correctly in your body to do whatever task it needs to do, whether it's an enzyme or a structural protein or it's part of something like the sodium-potassium pump. It needs to have the correct 3D structure in order to function appropriately. And once it does that, it will be a functional protein that will be able to carry out the task in your body. So please make sure you get all these notes copied down onto your Cornell note sheet. Bring all your questions with you. I know this is a very difficult topic, so you are going to have questions, and that is totally fine. I posted an additional video for you to watch that shows a live action version of how this process takes place. Please watch that video, and we'll watch it again, and I'll show you a little an animation on the board of how this actually takes place. But it's really important that you have all the notes copied down so you're ready to take additional notes and kind of just absorb how this works in class. Okay, so watch the other video on haiku and bring all your questions with you. Have a nice day, girls.